Hello and welcome to your ultimate guide for the HESI A2 course. My goal today is simple: give you everything you need to fully understand the key concepts, master them, and walk into your HESI A2 exam with total confidence. In this video we'll cover biological foundations, water chemistry, biological macromolecules, metabolic pathways, enzyme function. With over 100 plus success stories, we have proven our videos will help you pass your exam on the first attempt. And remember, our website offers free exam guides and practice questions to help you ace your HESI A2 exam. You'll find the link in the description below, so be sure to check it out and get the resources you need to study smarter, not harder. So let's get started.
Welcome to your complete guide to mastering the first half of HESI A2 Biology. Today, we cover everything you need to pass with confidence, including biology basics, the chemistry of water, the 4 major macromolecules, and metabolism. By the end, you will feel ready to walk into the test room. This video is built around the official HESI sub-objectives, so every concept is one the test writers may put in front of you. We move from the characteristics of life and the scientific method into water chemistry, then macromolecules, and finish with enzymes. Each section gives you definitions, examples, exam traps, and memory aids. Before we go deeper, I want to tell you about something valuable for your prep. I have over 300 free practice questions waiting for you in the description below. They are written in the same style as the real HESI A2 Biology section. Bookmark that link now and drill these concepts after the video. Let us start with the foundations. Biology is the scientific study of living organisms and their interactions with each other and the environment. The word comes from Greek roots, meaning life and study. Every nursing class you take, from anatomy to pathophysiology, grows out of these foundations, so a strong start here pays off. Living systems are organized into 13 levels, smallest to largest. We begin with the atom, then the molecule, the macromolecule, the organelle, the cell, the tissue, the organ system, the organism, the population, the community, the ecosystem, and finally, the biosphere. Each level emerges from the level below it. What makes something alive? Living organisms must show all the following characteristics. They have cellular organization, perform metabolism, maintain homeostasis, grow and develop, respond to stimuli, reproduce, pass on heredity through DNA, and evolve over generations. The HESI loves to ask which item from a list is not a characteristic of life. 2 of these deserve a closer look. Homeostasis is the maintenance of stable internal conditions despite outside changes, like body temperature near 37 degrees Celsius. Metabolism is the sum of every chemical reaction in your body. It splits into anabolism, which assembles complex molecules, and catabolism, which cuts molecules apart for energy. 2 more characteristics are reproduction and heredity. Organisms must produce offspring, sexually with 2 parents or asexually with 1. Heredity means each offspring receives DNA from its parents and inherits traits. Populations also show evolution as gene frequencies shift over generations. Adaptation is any inherited trait that boosts survival in a given environment. The scientific method is the orderly path used to investigate the natural world. The classic steps run as follows. Make an observation, ask a testable question, propose a hypothesis, predict an outcome, perform an experiment, collect and analyze data, draw a conclusion, and communicate your result. The HESI tests this exact order. Here is a distinction students always miss. A hypothesis is a tentative testable explanation. A theory is a well-supported, repeatedly confirmed explanation, like cell theory or the theory of evolution. A law describes a consistent universal phenomenon, like the law of gravity. A theory does not become a law. Theories explain why, laws describe what. Experimental design hinges on identifying variables. The independent variable is what the experimenter manipulates, plotted on the x-axis. The dependent variable is what gets measured, plotted on the y-axis. The phrase DRYMIX reminds you that dependent goes on y and manipulated independent goes on x. You also need the difference between groups. The control group does not receive the experimental treatment and gives you a baseline. The experimental group receives the treatment. A placebo is an inert version given to control groups. A double-blind study hides group assignments from both subjects and researchers, eliminating expectancy bias. Moving on to classification. Taxonomy is the science of naming and grouping living things, founded by Carolus Linnaeus. The hierarchy from broadest to most specific runs domain, kingdom, phylum, class, order, family, genus, and species. The classic phrase is "Dear King, Philip came over for good soup." Species is the most specific rank. Above the kingdoms sit three large domains. Bacteria and archaea are both prokaryotic, meaning their cells lack a true nucleus. Eukarya contains all eukaryotic life, including animals, plants, fungi, and protists. Bacteria and archaea differ in cell wall and membrane chemistry, so the test will sometimes ask you to recognize them as separate domains. Eukarya plus the prokaryotes are sorted into 6 kingdoms. Archaebacteria and eubacteria cover the prokaryotes. Protista holds mostly unicellular eukaryotes like amoebas and paramecia.I. Fungi are heterotrophs with chitin walls. Plantae are multicellular autotrophs with cellulose walls. Animalia are multicellular heterotrophs without cell walls. Notice that fungi are not plants. A favorite HESI trap concerns viruses. Viruses are pieces of DNA, or RNA, wrapped in a protein coat. They cannot reproduce alone, cannot maintain homeostasis, do not perform metabolism, and do not grow. For these reasons, viruses fail the basic characteristics of life and are not placed in any kingdom of living organisms. What we just covered is the kind of foundation material the HESI loves to test, and it is the type of question you will find in our free practice question bank. Those questions are designed to drill you on the same definitions, distinctions, and traps we are reviewing right now in this video. Now, let us move into water, the molecule of life. Water makes up roughly 60 to 70% of your body weight, and it is essential for nearly every biological process. The HESI A2 always includes water property questions because they show up later in fluid balance and acid-base disorders that nurses manage. Water has a special structure. One oxygen atom binds to 2 hydrogen atoms, forming a bent molecule with an angle near 104.5 degrees. Because oxygen pulls shared electrons more strongly than hydrogen, oxygen carries a partial negative charge while each hydrogen carries a partial positive charge. Polarity gives water its hydrogen bonds. The slightly positive hydrogens of one water molecule attract the slightly negative oxygens of nearby water molecules. Each hydrogen bond is weak, only about 5% the strength of a covalent bond, but together they explain almost every property of water that matters for biology. Cohesion is the attraction between water molecules of the same kind, which is why droplets form and water rises through xylem in trees. Adhesion is the attraction between water and a different substance, which is why water climbs the walls of a glass tube. Surface tension is the cohesive skin on water surfaces. Water also resists temperature change. It has a high specific heat, meaning a large amount of energy is required to raise its temperature because hydrogen bonds must break first. Water also has a high heat of vaporization, which is why sweating cools your body. Evaporating sweat carries off significant heat from skin. 2 more critical properties. Ice floats, because hydrogen bonds in solid water form an open lattice that spaces molecules farther apart, lowering density. Floating ice insulates lakes so aquatic life survives winter. Water is also called the universal solvent because its polarity dissolves many polar and ionic substances, supporting nutrient transport in blood. Water also forms different kinds of mixtures the HESI may test. A solution has tiny dissolved particles like saltwater or glucose in plasma.is. A colloid has mid-sized suspended particles that scatter light slightly, like blood plasma proteins or milk. A suspension has large particles that settle out, like sand in water. Polarity creates a key clinical concept. Hydrophilic substances are water-loving and dissolve in water, like sugar, salt, and amino acids. Hydrophobic substances are water-fearing and do not dissolve, like fats and oils. This sorting principle drives cell membrane structure, protein folding, drug solubility, and how the body distributes medications. The pH scale measures hydrogen ion concentration. 0 to 6 is acidic, 7 is neutral, and 8 to 14 is basic. Each whole pH unit reflects a tenfold change, so a pH of 4 is 100 times more acidic than a pH of 6. Human blood is held tightly between 735 and 7.45. Buffers are solutions that resist changes in pH. The most important one in your body is the bicarbonate buffer system. When acid builds up, bicarbonate binds the extra hydrogen ions, producing carbonic acid that breaks down into carbon dioxide, which the lungs exhale. The lungs and kidneys are the great regulators of blood, pH/DH. Memorize a few clinical pH ranges that the HESI may quiz. Arterial blood sits between 735 and 7.45. Stomach acid is around 1.5 to 3.5. Below 7.35 is acidosis, and above 745 is alkalosis. Both can be life-threatening. I know we are covering a lot of detailed information, and you might wonder how to practice applying these concepts. That is exactly why I put together those free practice questions. They are designed to help you work through problems that combine multiple concepts the way the real HESI A2 biology questions do. Now we move to macromolecules, the building blocks of every living thing. There are 4 classes you must know cold: carbohydrates, lipids, proteins, and nucleic acids. 3 of them, carbohydrates, proteins, and nucleic acids, are true polymers built from repeating monomer units. Lipids are not true polymers but are still grouped here. Polymers are built and broken by 2 opposite reactions. Dehydration synthesis joins monomers and releases 1 water molecule per bond. It is also called condensation. This is an anabolic reaction that requires energy. Think of it as removing water to create connection, which is why the body invests energy to assemble large molecules. The reverse reaction is hydrolysis. Here, a water molecule splits a covalent bond and breaks the polymer back into monomers. The phrase is that water lyses, or cuts, the bond. Hydrolysis is catabolic and releases energy. Digestion in your gut is hydrolysis in action, breaking starches into glucose and proteins into amino acids. Carbohydrates are sugars and starches built from carbon, hydrogen, and oxygen in a 1:2:1 ratio. Their monomer is the monosaccharide, and their polymer is the polysaccharide. They serve as the body's main short-term energy source. Glucose is the principal blood sugar that powers cellular respiration in nearly every cell. Carbohydrates come in 3 structural classes. Monosaccharides are single sugars like glucose, fructose, and galactose. Disaccharides are pairs, including sucrose, which is glucose plus fructose, lactose, which is glucose plus galactose, and maltose, which is 2 glucose units. Polysaccharides are long chains of monosaccharides linked into storage molecules or structural fibers. Distinguish the major polysaccharides. Starch is how plants store glucose. Glycogen is how animals store glucose, mostly in liver and muscle. Cellulose is the structural fiber in plant cell walls and provides dietary fiber that humans cannot digest. Chitin is the structural polysaccharide of fungal walls and arthropod shells. Each plays a different role. Lipids are diverse hydrophobic molecules made mostly of carbon and hydrogen with some oxygen. Major categories include triglycerides, which store long-term energy, phospholipids, which form cell membranes, steroids like cholesterol, and the sex hormones, and waxes that waterproof surfaces. Lipids do not have a single repeating monomer, so they are not true polymers. Phospholipids deserve special attention. Each one has a hydrophilic phosphate head and 2 hydrophobic fatty acid tails. In water, they spontaneously arrange into a bilayer with heads facing the watery environment and tails facing inward. This phospholipid bilayer is the structural basis of every cell membrane and is a heavily tested HESI concept. Fats come in saturated and unsaturated varieties. Saturated fats have no double bonds and are solid at room temperature, like butter or lard. Unsaturated fats contain 1 or more carbon-to-carbon double bonds and are liquid at room temperature, like vegetable oils. Trans fats are artificially hydrogenated and the most clinically harmful, raising bad cholesterol. Proteins are the most functionally diverse macromolecules. They are built from 20 different amino acids linked by peptide bonds, formed through dehydration synthesis. Each amino acid contains a central carbon, an amino group, a carboxyl group, a hydrogen atom, and a unique side chain called an R group that gives the amino acid its identity. Proteins fold into 4 levels of structure. Primary is the linear sequence of amino acids. Secondary is local folding into alpha helices and beta-pleated sheets held by hydrogen bonds. Tertiary is the overall 3-dimensional shape produced by R group interactions. Quaternary appears only when polypeptide chains join, like the 4 chains of hemoglobin. When heat, extreme pH, or certain chemicals disrupt protein shape, the protein is denatured and loses its function. A cooked egg is the classic example, where transparent egg whites turn solid as their proteins unfold. Proteins serve as enzymes, structural fibers, transporters like hemoglobin, hormones like insulin, antibodies, and the actin and myosin of muscle. Cholesterol gets a bad reputation, but it is essential to life. Cholesterol is a steroid lipid built from 4 fused rings. It regulates fluidity of cell membranes, serves as the precursor to all steroid hormones including testosterone, estrogen, progesterone, and cortisol, and is also the starting material for bile acids and vitamin D. Nucleic acids store and transmit your genetic blueprint. Their monomer is the nucleotide, made of a 5-carbon sugar, a phosphate group, and a nitrogenous base. The 2 types are DNA and RNA. DNA uses deoxyribose, is double-stranded, and pairs adenine with thymine and cytosine with guanine. RNA uses ribose and replaces thymine with uracil. The bases come in 2 structural groups. Purines have 2 rings and include adenine and guanine. Pyrimidines have 1 ring and include cytosine, thymine, and uracil. The phrase pyrimidines are cut helps you remember the single-ringed bases. Phosphorus uniquely identifies nucleic acids and ATP among the macromolecules, a classic HESI giveaway clue. One last comparison to lock in. Carbohydrates and proteins both yield about 4 kilocalories per gram. Lipids yield about 9 kilocalories per gram, more than twice as much, which is why high-fat foods are so energy-dense. Alcohol carries about 7 kilocalories per gram. These numbers explain energy storage and appear in HESI questions. If some macromolecule details feel overwhelming, that is completely normal. It is exactly why I created that comprehensive practice question bank. Working through those problems will help you sort glycogen from cellulose, peptide bonds from glycosidic bonds, and DNA from RNA. Practice makes these distinctions automatic on test day, and the questions are entirely free. Now we enter metabolism, energy, and enzymes. Metabolism is the sum of every chemical reaction in a living organism, organized into pathways. Anabolism assembles complex molecules from simpler ones and requires energy. Catabolism breaks complex molecules into simpler ones and releases energy. Anabolism assembles, catabolism cuts apart. Together they keep your cells running. Two laws of thermodynamics frame metabolism. The first law states that energy cannot be created or destroyed, only transformed. The second law states that every energy transfer increases the disorder or entropy of the universe. Living organisms are highly ordered, so they must constantly take in energy from food or sunlight to maintain order. The cell's immediate energy currency is adenosine triphosphate, or ATP. Each ATP molecule consists of an adenine base, a ribose sugar, and 3 phosphate groups linked by high-energy bonds. When the bond between the second and third phosphates breaks, ATP becomes ADP plus an inorganic phosphate, releasing energy that powers muscle contraction and nerve signaling. Enzymes are biological catalysts, almost always proteins, that speed up reactions without being consumed. They lower the activation energy of a reaction so it can proceed at body temperature. Enzymes are highly specific for their substrate and are reusable. Most are named with the suffix ase, like amylase digesting starch and lipase digesting fats. Each enzyme has a special pocket called the active site, where the substrate binds. Two models describe this fit. The lock and key model imagines a precise, rigid match. The induced fit model is more accurate and says the active site flexes slightly to mold around the substrate. After the reaction, products release and the enzyme resets. Several factors influence enzyme activity. Each enzyme has an optimal temperature near 37 degrees Celsius in humans and an optimal pH that matches its environment. Pepsin works at pH around 2 in the stomach, while trypsin works at pH around 8 in the small intestine. Substrate concentration, enzyme concentration, and inhibitors also matter. Two main types of inhibition show up on the HESI die. Competitive inhibitors resemble the substrate and bind directly in the active site, and you can outcompete them by adding more substrate. Non-competitive or allosteric inhibitors bind a different site and reshape the enzyme so the substrate cannot fit. You cannot overcome non-competitive inhibition this way. Many enzymes need helper molecules. A cofactor is an inorganic helper, often a metal ion like magnesium or zinc. A coenzyme is an organic helper, frequently derived from a vitamin, like NAD+ from niacin or FAD from riboflavin. Nutritional deficiencies cause specific metabolic problems because without the coenzyme, the enzyme cannot work. Cells regulate themselves through feedback inhibition, also called end product inhibition. The final product of a metabolic pathway acts as an allosteric inhibitor of an early enzyme in that same pathway. When enough product builds up, the pathway shuts itself off. Once the product is used up, the brake releases and the pathway resumes. Mastering these concepts takes practice and application. The free practice questions are designed to help you take what you are learning and turn it into exam-ready skills. They include the same enzyme inhibition scenarios, ATP questions, and macromolecule identification items you will see on your real HESI A2 Biology section. Speaking of enzymes, the HESI sometimes asks you to match digestive enzymes to substrates. Salivary amylase begins starch digestion in the mouth. Pepsin breaks down proteins in the acidic stomach. Pancreatic amylase, trypsin, and lipase finish carbohydrate, protein, and fat digestion in the small intestine. Lactase, sucrase, and maltase break disaccharides at the brush border. Let me flag a few common HESI traps so you can avoid them. A theory does not become a law. Cohesion is between same molecules. Adhesion is between different ones. DNA contains thymine while RNA contains uracil. Lipids are not true polymers, and viruses are not classified as living. These distinctions catch students every year. Now let us lock in the most useful mnemonics. Dear King Philip came over for good soup gives you taxonomy. DRYX reminds you which variable goes on which axis. Pyrimidines are cut names the single-ringed bases. Anabolism assembles. Catabolism cuts apart. Use these phrases to anchor memory under exam pressure. For test day strategy, remember you have about 50 seconds per biology question. Read all 4 answer choices before selecting because the HESI loves best answer items where 2 options seem correct. Eliminate obvious wrong answers first. Watch for absolute words like always or never, which are usually wrong, and trust your first instinct. As we near the end of this overview, remember that understanding these concepts is just the first step. Real mastery comes from applying them, which is exactly where those free practice questions become invaluable. They are completely free, organized by topic, and designed for HESI A2 success in nursing program admissions. Click the link. Let us bring it all together. We started with foundations like levels of organization, the characteristics of life, the scientific method, taxonomy, and how viruses fail those criteria. Then we explored water, where polarity and hydrogen bonds create properties like cohesion, adhesion, surface tension, high specific heat, ice flotation, and universal solvent capacity. From there, we moved into macromolecules, where dehydration synthesis builds and hydrolysis breaks. We covered carbohydrates for energy, lipids for membranes and storage, proteins for structure and function, and nucleic acids for genetic information. Finally, we tied it together with metabolism and enzymes, where ATP fuels life and enzymes lower activation energy. Notice how everything connects. Polarity from water explains the phospholipid bilayer of cell membranes. Hydrogen bonds stabilize protein folding and DNA base pairing. Enzymes are proteins that depend on shape, which depends on amino acid sequence. The HESI tests these connections, not isolated facts. Walk in knowing the story of life from atoms up. You have built a strong foundation today, but practice will turn that knowledge into points on the test. Do not forget to grab those completely free practice questions from the link in the description. With everything we covered today plus those questions, you will be incredibly well prepared. Good luck, future nurse.